Hi, welcome back. In this video, I'd like to talk to you about data structures and conventions in genomics and bioinformatics. It's very important to keep in mind that when we're doing bioinformatics, we're really using computers to interact with data, genetic data, and that requires that we, in order to communicate those things effectively and analyze them effectively, that we follow some strict conventions for that. Basically, bioinformatics really relies on the use of common file formats, because when you write programs, there have to be predictable ways of knowing where and how to expect data to appear. So one of the simplest concepts in genetics, and one of the great observations in biology, is the structure of the double helix of DNA. And one of the things that we know from this, and is explicit in the quote from Watson and Crick's original paper, is that one strand of a double helix actually contains all of the information to specify its partner strand. Okay, and so in bioinformatics, what that means is that we don't need to write both strands of a DNA molecule out. We can provide the information for a DNA sequence with a single strand of DNA. And by convention, when we communicate that to someone, we always depict that in the 5' to 3' direction. Okay? And if we need to know, and we very often do, the complementary strand, it can be produced informatically from that without error. So one of the most important and common formats for depicting DNA, or in fact protein sequences, is what's called the Fasta file format. So Fasta files, the name Fasta file is really a legacy from some early protein sequence alignment software called Fastq, in fact written by Littman and Pearson back in 1985. And so the name Fasta file comes from that data structure. But Fasta files are one of the most common ways that we use to communicate DNA and protein sequences. And their structure is very important. The structure of a Fasta file is such that if we look at this nucleotide Fasta here, the first line in a Fasta file always begins with this greater than symbol. And on that line that begins with that symbol is a unique name or header. And that header is the unique name for the sequences that follow on one or many thousands, hundreds of thousands of lines of sequence that follow that. And all of those sequences fall under that one name and are on our one DNA strand that begins at the 5' to 3' end. Similarly, on the protein end, you also have a header, begins with a greater than symbol. All the information that you feel necessary can be in that header for each of these protein sequences, and it goes from the end to the C terminus by convention. Okay. Less assured, but what we'll talk about later on in this course is that these files, so for example, when you have Fasta files, they will have extensions. So like a text, like a Word document would be .doc. Well, these have 3-letter extensions, or sometimes more than 3-letter extensions. And these are generally followed, but not always strictly followed, but are very important ways of understanding before you look at a file or use a file exactly what might be in there. What's depicted here at the bottom of this is another Fasta file, and what often happens, less so, we'll talk about other ways that quality scores are presented to you, but it's very often true that when you have a DNA sequence that you also want to know something about the quality of that DNA sequence. And so, for example, what we have down here is a file that's parallel with this. So every single nucleotide in this nucleotide Fasta has a corresponding numeric score, which are the Fred quality scores, and tell us something about the probability that when that T is actually a T and not an A, G, or a C. And so those quality scores are meant, the numeric value of those, or the Fred quality score, which is meant to give you some sense of the probability that that base is correct or incorrect. And so a quality score of 10 simply means that there's a probability of about 1 in 10 that that is wrong. And so we generally don't think that's very good. Whereas a quality score of 30 tells us that, or suggests to us that there's about a 1 in 1,000 chance that that base is called incorrectly. And so for every sequencing technology, there's different algorithms for predicting the Fred quality scores, and there's lots of information out there on this, but that's generally how they're used. Where you will see quality scores more often now, and you will see them when you receive your data, is in another kind of file format that carries on that naming strategy, and that's the Fastq format, which really the Q stands for quality, because in these cases the sequences and their qualities are actually kept together in the same file, rather than being in two parallel file sets. So in the Fastq format, just like in a similar way to the Fasta formats, there's a header line, or the title of the sequence that is to follow, and in this case it begins with an @ symbol. Okay, so every new sequence is going to begin with a line that has an @ symbol in it. And then that's followed by sequence lines, and that's the same right here, you can see. And so this is a typical Fastq file. And then there can be an optional repeat of that original title. It's optional. And then that's followed again by a parallel set of quality scores. And you might be going, wow, those do not look like Fred quality scores, but they're actually just the ASCII representations, the single character representations of those same quality values. Okay, and so this brings up a couple of points. And one of them is the fact that part of the reason for doing this is that this is the way next generation sequencing data is transmitted and stored and presented. And so cutting down on the amount of space is very important. And so the compression of these files is really critical. And so that's part of the reason for doing this. The other is that these files, just by their very nature, are not meant to be read by humans. I mean, we will show you how to read your Fastq files and to look at them, but the idea that you would go through 100 million lines of text by eye is not realistic. And so the fact that they're in some terse and crude way of representing numbers is not a problem. So there are some other conventions. We all know about the G, A, T, and C definitions for the nucleotides in DNA, and the U that's found in RNA, although we almost always depict it as a T for whatever reason, because we're lazy, I guess. But then there are other degenerate versions of this. For example, you may be familiar with the idea that we often can represent either of the pyrimidines, the T or C with a Y, or the purines with an R. And then there are the other designations that are single letter as well. The most common one that you'll come across in Fasta files and sometimes Fastq files is an N, which just usually reflects the fact that the quality is too poor to designate a specific nucleotide. And then there are similar single letter representations for the amino acids in protein sequences. I just want to talk briefly about file formats that are common. There's a couple of file formats that are common and that you will run into as part of your project here. One of them is a GFF file, which has various names: general feature format, generic feature format, whatever, and it's the file extension is GFF. Basically what this is a way of describing where the genes are coded in a FASTA file. So you have let's say you have a bacterial chromosome in a FASTA file that may be millions of nucleotides long, and you figured out, and you will do this as part of this this project, you want to be able to describe where a protein coding gene starts and stops. Okay, and so we can do that in a format like this, where, for example, in a line of text with information separated by spaces, you in the first position you say what sequence you're working with. Well, okay, so that's chromosome 1 from this organism, which was the second feature there. And then we can say, well, all right, so what am I talking about? Well, I'm talking about a protein coding sequence. And so we often call those CDSs. So it'll say CDS in the third position. And then, so where does this start? Well, it starts at position 6,482 and ends at some number bigger than that. And the score for that kind of sequence, the quality of it is this. And oh, by the way, this is on the strand that is depicted here, or alternatively the complementary strand, and that the amino acid sequences are read in the first frame, and what other attributes it has there. And so the important thing about this, and you'll see these, is that this information is always in the same place and of the same format. And so if we're going to then use or interpret a GFF file, then every GFF file will have the information in the same place, and that's what's really important about those. Another sequence file that has an important format that's very common in different applications in bioinformatics and genomics is what's called the SAM file. It's a very common output for assembly or alignment files, and it's the sequence alignment map. And basically what this file is, again, it's just a text file with the information in predictable places separated by spaces. And it just tells us if again you have a bacterial chromosome or any kind of a FASTA sequence, and you have compared or aligned one of your sequences to it. So let's say you take some of the sequences that you have and you want to look and see where those reads come from in a genome. Well, that's what you'll end up with then is a SAM file, and there's a binary version of it which is not human readable, but is much more preferable for programs to read, which is called the BAM file. And so those, again, it's a similar treatment, because what matters here is that the information is of the same type and it's in the same positions, predictable positions. So, for example, the first thing is what's the thing that you're mapping to the genome? What's the name of the read? And so you have something that is meaningful in terms of what the read name is. And then there's some what in this case it's called a bitwise flag, which for the programs that use this is a important piece of information that you'll learn about if you need to. And then it talks about, well, what's the reference that you're mapping to? So it's a sequence alignment map. Well, in this case, it's chromosome 1. And so maybe this is chromosome 1 from a human. Maybe it's chromosome 1 from Drosophila. Maybe it's chromosome 1 from C. elegans. I don't know. We could probably check. And then where is the start position of the sequence that's aligning there? And so this tells us it's 497. And then we carry on with the quality of the map, which is 37, which simply means that every single nucleotide in that sequence matched. And that's actually reflected in what's called the cigar string, which tells you that that every nucleotide matched again. Of course, if it didn't, it would also tell you which matched and which didn't. And then it tells you where the other pair of a sequence mapped, and you can see that that's not even on the same chromosome. And then it gives you the sequence, the 37 nucleotides of the read that you mapped, and it tells you about the quality of that read there below. So the important thing here is you don't need to memorize what these are, but that's the structure, and every SAM file has to have the exact same structure. And when you generate it, it will. And when you use those files, they're useful because they're in that structure. So thank you very much, and I look forward to talking to you again soon. Thanks.